Assalamu alaikum dear students. Today we will be talking about classification of fungi. The kingdom fungi, it has been divided into different classes on the basis of some important criteria, and these criteria include morphology of mycelium, how mycelium looks like, is it synostic or it is septate. Okay. Second one is mode of spore formation. What are the types of spores produced? And the third one is fruiting bodies. Okay, so these three criteria have been utilized to classify fungi into different classes. The first one is, I will repeat, morphology of mycelium. What is the type of mycelium? Then mode of spore formation, whether spores are produced endogenously or exogenously, and the fruiting bodies. What are the types of fruiting bodies in the fungi? And on the basis of these three criteria, we have classified fungi into four classes mainly. The first one is Ficomycetes, which are also known as larva fungi. Then Ascomycetes, also known as sac fungi. Basidiomycetes, also known as club fungi. And finally, Deuteromycetes, which are also known by the name Fungi imperfecti. So today we'll be talking about the first two, Ficomycetes and Ascomycetes. So let's move on first of all with the Ficomycetes. Ficomycetes, they are mostly aquatic. Some are known to live on decaying wood in moist and damp places, while as others, they are known to exist as parasites on plants. The mycelium in case of Ficomycetes, it is aseptate and multinucleate. Aseptate multinucleate means there is no intervening septa. Septa are absent, okay? And these hyphae, they are multinucleate. This aseptate multinucleate condition, I have already told you, this is known as synostic condition. So simply stated, you can say they are synostic. Now they are asexual reproduction. Asexual reproduction in case of Ficomycetes, it occurs by the formation of both motile zoospores and non-motile aplanospores. Okay, in some species of Ficomycetes, zoospore formation takes place, while as in others, aplanospores are produced, non-motile spores are produced. Okay, now the mode of spore formation. The spores in case of Ficomycetes, they are produced endogenously inside the sporangium. Here this is the sporangium, structure of sporangium, and inside the sporangium many spores are produced. So the origin of spores is they are endogenously produced. Now there is sexual reproduction. Sexual reproduction takes place by a number of methods, and these methods may be either isogamy, anisogamy, or ogamy. Okay, I will tell you what is isogamy, anisogamy, and ogamy. We have already discussed these things. So in sexual reproduction, there actually takes place the formation of a zygospore, and this zygospore formation occurs when two cells they fuse, their contents fuse, and you will get the zygospore. And this zygospore, it is diploid. So zygospore formation can occur by either of the three methods. The first one is isogamy. In isogamy, the fusing gametes, they are similar in their morphology, they are similar in terms of size, they are similar in terms of behavior. Then we have anisogamy. In anisogamy, the gametes are dissimilar. They may be dissimilar in terms of their size, they may be dissimilar in terms of their behavior. Okay, and finally, oogamy. In oogamy, we have large non-motile egg and small active sperm. Sperm is active, while as egg is non-motile. Okay, the examples of fungi in this class, it includes mucor, rhizopus, albigo. Okay, and this albigo, it is also known, it is known to cause disease in mustard. It is a parasite of mustard. Okay, so these are some representative examples of Ficomycetes. Now the second class is Ascomycetes. Ascomycetes, they are also known as sac fungi because the sexual spores in these species, they are produced in sac-like structures which are known as SI, or you can say Ascus is singular and si means plural. Okay, so those fungi in which sexual spores are produced in sac-like structures or sac-like structure ascus, they are known as ascomycetes. Ascus is simply a sac-like structure, and inside the sac-like structure ascospores are produced. Usually 8 ascospores are produced. They are mostly multicellular. The ascomycetes, they are mostly multicellular. However, there are some rare examples where the ascomycetes they have unicellular body. Example is yeast. Yeasts are unicellular. Most of the ascomycetes, example penicillium, they are multicellular, but some ascomycetes, example yeast, they are unicellular. The ascomycetes, they are saprophytic decomposers or coprophilous. Saprophytic decomposers means they obtain the organic food material through their general body surface by causing decomposition of the biomass. Okay, they are saprophytic decomposers. They decompose the organic matter and obtain the nutrients from it. Or you can see some of these ascomycetes, they love to grow on dung. Coprophilous. Okay, coprophilous means dung loving organisms. The mycelium in ascomycetes, it is branched and septate, and the septa, they are simple pored. I have already told you what is simple pore and what is roly pore. Okay, the mycelium is branched. Here you can see the mycelium is branched and septate. You can see these are the different septa. They have divided the hyphae, the mycelium, into small segments. Asexual spores, which are also known as conidia, they are produced exogenously. This is the difference. The asexual spores in case of ascomycetes, they are produced exogenously, and these asexual spores in case of ascomycetes, they are known as conidia. Okay, here you can see this is the structure. You have here this is known as conidiophore, and on this conidiophore you will have conidia in the form of chains. You will have conidia, a chain of conidia here. These represent the chains of conidia which are formed. Now the sexual spores. Sexual spores, which are also known as ascospores, they are produced endogenously in ascus. Here, this is the fruiting body of ascomycetes. Okay, and inside this fruiting body you will have structures which are known as asci, and single structure is known as ascus. The sexual spores in ascomycetes, they are produced in these asci. Okay, but endogenously. Endogenously means they are internally produced. Now the ascocarp. Ascocarp is the fruiting body which is produced in ascomycetes, and these fruiting bodies, as I have already told you, they contain the asci, and inside asci usually 8 ascospores are produced. Yaad rakhna, I will show you here. This is the ascus, and inside this ascus you will be having 8 ascospores. So these structures, these represent the ascocarpus fruiting bodies, and inside these fruiting bodies you will have many asci, and inside each ascus usually 8 ascospores are produced. Okay, now the examples of fungi representing ascomycetes, we have we have many examples, but here we will just quote a few. The first one is aspergillus. Then we have Clavicepus and Neurospora. These are some representatives of ascomycetes. Many members like morels and buffaloes, they are edible and are used as delicacies. Okay, So these members, morels and buffaloes, which belong to Ascomycetes, they are edible and are used as delicacies. So this was all about the Ficomycetes and Ascomycetes. In the next part, we are going to talk about Basidiomycetes and Deuteromycetes. Till then, stay blessed and stay safe.